Hello and welcome to Rajat TV. You're watching To The Point with the host Rajat Kain. Well, as world continues to reel under the COVID-19 pandemic, there have been several studies to understand this virus better for vaccination as well as treatment protocol. In short, how to fight the menace of COVID-19. At the same time, there has been a renewed clamor so as to trace back the origin of COVID-19 pandemic. There have been some studies conducted under WHO, and of course there have been independent studies and research as well based on several hypotheses as to how this virus could have possibly originated. In India, there is a scientist couple based in Pune who have conducted their own independent study and research, and of course what are the hypotheses, what were the findings, and how they reached to this conclusion, if at all this is a conclusion. We're joined with Dr. Monali Rahalkar. She is scientist, Agharkar Research Institute, Pune, and Dr. Rahul Bahulikar, scientist, BAIF Development Research Foundation in Pune. Well, many thanks, Dr. Rahalkar and Dr. Bahulikar, for joining us. Indeed, pleasure. And of course, it's an esteemed honor to have both of you in the panel who have conducted this study, so as to say, or the research tracing the origin in your hypothesis of COVID-19 pandemic. Let me start with you, Dr. Rahalkar, Dr. Monali Rahalkar, ma'am. To begin, to make our viewers understand as to what was your study, let's see, we will of course walk backwards, but what has been the finer findings of your study? Let's start with the finding. We'll of course go backward, ma'am.
Yes, we started to find more about COVID-19 virus origins. We started in around April 2020.
Okay.
And as you said, I will list the major findings of our study. What we found out was that in 2012, there was a mine in south of China in the Yunnan province. It was a copper mine, and in that abandoned copper mine, it was a bat-infested mine, right? So there were several kinds of bats, and there were 6 people who went to clean the bat feces, and they got infected with a lethal pneumonia. Like 3 of the miners who had just went there for cleaning the mine, they died out of it, and they were hospitalized in the same hospital in China. Now, Wuhan Institute of Virology was conducting studies on bat coronaviruses on this particular mine because of this incident, and they were visiting the mine for 3 years. Our study documents that we had got a very important reference from the Chinese official website. It is a master thesis published by a medical student. So it was a very authentic study. And we compared that particular pneumonia cases with the present COVID-19. Our findings clearly indicate that the major symptoms, the minor symptoms, and the treatment was very similar to COVID-19.
Okay.
The second point is that the closest neighbor of SARS coronavirus 2, which is called RATG13 virus, this sample was also collected from the same mine shaft in Mojang.
So basically, this Pune couple of Dr. Monali Rahalkar and Dr. Rahul Bahulikar, sir, as I asked Dr. Rahalkar, same question to you, sir. What was the inspiration that led both of you to work in this, or rather delve deeper into this mystery?
Okay, when the last lockdown started, that time we were curious to know why this is what is happening and why it is happening. Further, the information getting from China was very scanty. Means some information like only they inform us when the pandemic was at the peak, and before that there was no mention about it. So that was a kind of anger, and then when our close relatives are getting infected with that, it was like giving us a little bit anger. And as a scientist, there was a thirst to know what's happening at the level of where from this virus is coming. Either it has zoonotic origin, or it is a lab leak, or something else. So we after that we wanted to started to digging out about it, how and where from it started.
Right, right. Dr. Monali, there is an important dimension to the hypothesis that you're giving here. Ma'am, as you said that this possibly is, I mean, as per your hypothesis, this is not a naturally grown virus, whereas rather it seems to be more of a lab leak here. There has been several theories about this aspect earlier as well. How is your study different from theirs? If at all it is different, or rather it's adding any value to that hypothesis?
Okay. Our study basically focuses on this particular virus called RATG13 and its origin. So RATG13 originated from this bat-infested mineshaft, right? Our hypothesis is that Wuhan Institute of Virology collected several samples from this mineshaft, and actually 8 more SARS-like coronaviruses from the mineshaft collected, and it is a completely different clade of viruses if we compare it with the SARS-1, which was originated in 2003. Now, looking at the expertise of the WIV, it is very clear that they can create chimeric viruses using backbone of a particular virus and spike of another virus. So our hypothesis is that the RATG13, which was the next relative of SARS coronavirus 2, could have been used as a backbone or a backbone sequence, to maybe artificially, either synthetically generate the virus, or maybe there were certain changes made to this RATG13. This is our hypothesis. And maybe they did it for certain experimental reasons,
right.
But due to some human error or, less security, it got out. This could be this is our hypothesis.
Mm-hmm. And how difficult was to get access to the information, especially coming from, Chinese sources or information which was based out of China, ma'am? Given the censor and given how they curtail information over there in a lot of Chinese publications. So how difficult or challenging was it to get a lot of research data that you found so far or studied so far? How difficult was the access, ma'am? Please.
Yes, actually, we published a preprint initially based on our research, and luckily we were contacted by a reader who had read our preprint. He is a Twitter user, and there was already an unofficial group of scientists and other, you can say, open source intelligence people or architects, and it was a diverse group. So they were also working on this issue, and one of them, called Seeker, he contacted us. He sent us a master thesis, and this was on the Chinese official website. So I would like to tell you that there's a website called cnki.net. So it is an official website for depositing all the Chinese master and PhD thesis, and we could download the article without any problem by creating our own password and login.
Right, right. So what were your initial findings? I mean, were they startling? Did it move you to delve deeper into it? I mean, I just want to know your experience. Of course, it's really interesting as how you started off, and the findings, the final findings or conclusions of your hypothesis has given a new dimension to it. But initially, when you started with it, I mean, what were the challenges you faced?
Okay, about the paper which we have published. Basically, we first came to know that this origin of this virus came from a mine. Monali already mentioned some story about it, and it is 96% of the virus which is at the present. And further things are, even though the distance between these, like Yunnan, where the virus is found, and the Wuhan laboratory, it is more than 1,000 miles.
Right.
So, and this virus cannot migrate more than 25 kilometers. So as for the migration, it is too high distance. Further, whenever we also know that the samples were taken from the miners, and they have been sent to Wuhan Institute. So we wanted to know like what happened and the correlation between the present situation and present symptoms of the present patient symptoms as well as the minor symptoms. And in our manuscript, we have shown that these were highly similar.
Right, right. Ma'am, could you tell us more about, like, in terms of the sequencing of your research and hypothesis, starting from, that bat cave which you mentioned, and how it went on growing to become such a catastrophic pandemic throughout the world? Could you be a little more elaborate about it?
Okay, so these are just guesses, but I would like to tell you in short what was the work going on in such labs, because the world doesn't know, we do not know what is going on, in such labs. So initially the Wuhan labs, or especially WIV, they were focused on bringing this bat coronavirus samples. So these are present in the bat fecal matter of horseshoe bats, especially the SARS-like coronaviruses. So they have a large collection of bat coronavirus samples. Around 22,000 samples are there in their collection. After that, what they do is they try to sequence these particular samples using next generation sequencing. And after sequencing, what they do is they do different experiments. For example, if there is a live virus, for example, if they have a virus isolate, they can change the spike region by cloning the spike region into the backbone of the other virus, and then they check it in humanized mice. So what are humanized mice? So these are mice which express the human ACE2 receptors in their respiratory systems. So these mice behave like human beings, and when they get the disease, then they are sure that particular virus is infective.
Okay, okay. And then, like, did you find ever in your research about how it can vary in the severity? Like, I mean, this is more to do with like what we're facing in world right now, comparing the first wave that happened last year in 2020, and then second wave which we just experienced in India. It has been in particularly catastrophic.
There is a new publication also by a Norwegian and a British scientist. And what they state is that they also state that it could be, sort of a manipulated virus, because there are certain features in the virus which are very unique. For example, the receptor binding domain and the furin cleavage site. Now, this receptor binding domain has certain amino acid residue sort of patches which are positively charged, and they are very close to the receptor binding motif. Now, what happens because of this is the binding of the receptors to our human cells is very tight in this case, and that's the reason that SARS coronavirus or the COVID-19 pandemic was so severe, and the transmissibility of for example, the double mutant, it is more than the first mutant, because of this particular furin cleavage site getting more basic, or the particular structure of the receptor binding domain, I would say.
What about challenges, like how you laid your hands on the research material, and of course like literature or other, I mean, anything connected to Chinese literature, or to say Chinese domain, as to like it's rather very difficult to actually access anything which has any Chinese blueprint, sir?
Yeah, the thing is, from the beginning, we found that there are many discrepancies in it. Like the name of the virus, which was in the mine, that was mentioned RATG13, and the virus which were in some publications was 4911. So first we wanted to. It took us time to say that both the sequences of both these viruses were same. Then the information given by Zheng Shi, in some cases she was mentioning about the mine, some cases she was not, just we got some sample from somewhere. So there was also scantiness, and the publications, although we follow most of them publications, the information in some places was either of like some things are given and some things are not given. So it was again problematic. And based on the initial information when we published the first our reprint, and we tried to contact Jenglishi, but we also read the other people's opinion that they also did that and there was no information available.
Right.
After that, after we published this paper, we communicated this paper, and in that also after that, after publishing that paper, we after 1 month of that, Jenglishi published addendum for that, and in that like answer some of the questions but not all. Again, we wrote wrote a few points, and there was no answer. Like getting information from Jenglishi or any other people was like very difficult.
Very difficult. Okay. Right. Ma'am, as you mentioned that in your research you found that there were minors who had gone into the bat caves and they took ill. In terms of the symptoms that they had shown, how similar those symptoms were of any average patient suffering with COVID-19? How similar were the symptoms of those minors affected by, in the bat cave and COVID patients, so that like we can put 2 and 2 together?
Yes, I will tell the major symptoms. So in COVID-19, we have fever, cough, pneumonia, right? And mainly the difficulty in breathing. So the lung part is affected. So the minors had all these particular symptoms. Some of them had to be put on ventilators, I think 4 out of 6. And most important thing was that the CT scans of these minors showed ground glass opacities, which were of peripheral consolidation was seen. So we also consulted radiologists and we confirmed that these particular features were similar, the radiological features. Also the blood work, I mean the laboratory tests were very similar. So there was decline in lymphocytes, there were increased D dimers, and there were increased serum, this one marker called SAA, serum amyloid albumin. So this marker was also elevated. Plus these minors had other secondary infections like fungal infections, bacterial infections, which were very similar to seen in the COVID-19 patients.
Right. That's remarkable. And ma'am, as we move towards the close of the show here, question to both of you. Let me start with you, ma'am. We have seen there were several other forums or individuals who have come, who have been working on this. Most of them prefer, say, anonymity. But 2 of you have actually come out and really helped us in understanding the nature of virus. So how important do you think that it is there for anybody who's studying this virus or its sequencing or any nomenclature of this virus to actually come out with the study and show it to the world?
You mean the importance of coming out and,
Yes,
publishing such kind of studies?
Absolutely.
I think it is important to show this courage because I am a scientist working in the government lab, but we did this out of interest and we did it in our extra time, all our evenings and night time and whenever we had time. So we had this and there are other individuals who have devoted almost, 50 hours per day for this particular work without getting any benefit. Now we have published this research in peer-reviewed journal just because the world should not treat it as a conspiracy, and finding out the origins is very important because we should try to prevent the next pandemic. Otherwise we won't be able to understand, and if there is another pandemic we would just be left in darkness, if we do not understand why this happened. So it is important to publish papers, to give out this information in public, in the form of even preprints or articles. So I think this is very important kind of work in general.
Certainly, we have understood what Dr. Rahul Bahulikar was saying, and so was Dr. Monali Rahalkar. Certainly, there is a research that has been done on a very specific hypothesis of how the pandemic started, and more research on all other aspects of COVID-19 will definitely enrich our understanding of this disease, of this virus in particular, so as to have treatment protocol that can be effective in curbing the menace of COVID-19. Well, many thanks to Dr. Monali Rahalkar and Dr. Rahul Bahulikar for joining us from Pune and certainly throwing light upon your research and elucidating further as to what were the findings. Well, to our viewers, thanks for watching this edition of To The Point. Thank you and goodbye.